So what we're going to start with is, obviously, this is not the setting you would ever do an extraction like this, right? So you would do the extraction in ancient DNA lab or archive lab or historical lab, whatever it is, very, very far away from anything that is running PCRs, ideally in a different building. And you would also separate the steps of getting the powder out of bone or tooth or any type of material and the actual extractions. Because usually getting the powder out is quite a dirty process, it sprays stuff. First thing you do is you gear up, obviously. So this is like kind of a simple lab coat. Ideally, you would have a full body lab coat, right? That covers up everything. And I'm a big fan of cleaning, so 90% of the time I spend cleaning and gearing up. And what we usually do is we have, I think most people do, 2 layers of gloves. So one layer is going to be glued to the coat, and the second layer is free to change at every step of the protocol. And also what you would usually do, you would put this thing on, you would glue it back to your head, and you would put on a face mask. I'm just going to show you how it's supposed to look like, but I think I'm going to take it off because otherwise it will be a little bit difficult to speak. Ideally you have a shield to protect your eyes as well so that nothing falls out ever. And then you look like a spaceman. All right, so second layer of gloves, and this is what we work with, and that's what we can change very, very easily. Right? Okay, so, yeah, the samples. So we have, six samples to process. What you always do, you have to include blanks, right? Obviously. What we do regularly, depending on the number of samples, we put a blank in the beginning and a blank in the end. If you have more samples to process, you include a blank in the middle as well. Now we have what we did, thanks to Gonton and the museum, we have 2 teeth from 2 dog specimens. One of them is from 1876, and the other one is really modern from 2003. And what we did, from these 2 teeth, we extracted a cementum fraction, or a fraction that's enriched in cementum, so the outer layer of the root, and we grinded the dentin fraction, so the fraction that's inside the root. Sorry, I'll construct a something for the tooth to hold it into, to fix it on the surface, and we would put it like this with the surface from which we want to collect exposed and under cross-linking, under UV for 10 minutes.
This hand drill, and this is the most convenient you can actually find. So. One of the most important things is that it can drill at low speed. So this one can go down to 2000 RPM, which is relatively low for drills. So we advise to never go above 5000 RPM because you will smell this burning smell. You're actually burning your sample, and that's of course not what you want. So the most important thing is that it's convenient to use. So this is the preferred tool. And Gontran said they're not so. Is something here? No. So they are not so expensive, and they can drill at low speed. So yeah, this is what we would advise for the drilling. So I always use aluminium foil as a first layer, and then I have this. It's called the wetland paper. So it's sort of a wax paper. So it's a paper with a small layer that should not bind anything, so it's easy for collecting the powder. The first thing I will do is clean the outside, so just by removing the surface from one of the roots. And then I will change my drill pieces, and I will try to collect a clean layer from the outside of the root as much as I can. So this would be the cementum fraction. in this case, it's impossible to see where the cementum stops and where the dentin ends. So we sort of call it now a cementum enriched fraction, and the other fraction will be the dentin enriched fraction. And in our experience, and also in what's published in literature, the cementum seems to be the best part of the route for your DNA extractions. So I found out here that these drill pieces are very good to clean the surface. So that I had never used it before, just here, but it worked really great. And then you use the very, very small drill bit, so there the tip is like, it's incredibly small. It's always finding a good way of holding it. Yeah. Yeah, so I draw at low speed, so this is about 2000 RPM. So we have been actually drilling at quite a lot higher speed, and this still seemed to work, but if you can drill at lower speed, then obviously try to do it at lower speed. So I'm now just cleaning the surface basically. Also, the protocol says that you should aim for like a minimum of 30 milligrams. Ideally, you would have 50 milligrams. But if you have like a large sample, or like a sample where you can get a lot of material from, you could even think about like collecting 100 milligrams, for instance, and doing 2 extractions just to give you more material. But this depends on the size of your sample, how precious the sample is, how much you can actually are allowed to take from the sample. But try to get at least 30 milligrams of the sample, and I will try to show you like what volume corresponds to around 50 milligrams, and then you can sort of see.
Well, we have a scale here.
We have a scale, yeah. Yes. Yeah, so this is always a bit tricky. So that's in principle how you collect the outer layer of the tooth. Now it looks okay because we have really fine powder, so we will see like very small pieces of bone still, but if it's too much, what we have been doing is adding some extra proteinase K, so this is the enzyme that breaks down the bone powder. So we incubate overnight, we check the tubes, and if there's still a lot of powder left, we added some extra proteinase K and increased the temperature a bit and left it standing for another 3 hours. But like some larger bone fragments, they almost never completely break down. Yeah.
So the thing also is, you'll realize when we do the first part, we incubate the samples at 37 degrees. This is not the ideal temperature for proteinase K to work. Exactly. The reason we do that is because our DNA is fragmented enough, and it's precious material, and we don't want to subject the DNA to another strenuous, type of fragmentation environment, which would be hot temperature. So that's why we're extracting our samples at very low temperature to preserve the DNA as much as we can. So it's not for the sake of prot K, it's for the sake of DNA. So what we need to do is, this extraction buffer is based on the guanidine hypochlorite and sodium acetate. So we have both of these chemicals here. We have prepared the bundles. You see the buffer protocols are on the back of the first page. So the binding buffer has been prepared and it has been UV'd, so it has been decontaminated. It's actually quite interesting because usually tween 20 that we add to almost all of our buffers. So tween 20 is this detergent that prevents DNA from binding to the walls of the tubes. Frequently you probably have this experience that if you thaw and freeze your samples multiple times, the sample quality decreases. And the general explanation for that was that the DNA breaks down through freezing and thawing processes, but this is actually not what happens. What happens is that during freezing and thawing, DNA binds to the plastic walls. It adheres to the plastic walls of the vessel. So to prevent this from happening, you add tween 20. So it's a detergent that keeps the DNA in solution and at least prevents this adhesion of the DNA to the walls. So that's why we like using tween 20. But tween 20 itself is in principle light sensitive. So it shouldn't be exposed to light, and these guys should be kept in the dark. But we still UV it, and it has worked for us so far because it's a very nice way of decontaminating the buffer. And clearly, before you start working, you clean not just the surfaces, but also you clean your pipettes, right? Not with bleach. They're not going to be happy about this. If you have DNA away, that's the best way of doing it. So what I'm going to do now, I'm just going to add 400 microliters of sodium acetate, 3 molar. Is that right? Yes. pH 5.2 to each of these tubes before we actually do anything with our samples, right? So just okay, so now the 400 microliters of sodium acetate in here, and now we're gonna, because we incubated our samples in 1 milliliter extraction buffer. So if we did use more than that, for example, because your sample was larger and you wanted to add more extraction buffer, you'll have to adjust the volume of sodium acetate. So now we're gonna add a binding buffer to these guys, and we need to add 10 milliliters. As it says in the protocol, the ratio has to be at least 1 to 7 between the extraction buffer and the binding buffer. So we have 1 milliliter extraction buffer, so we need to add at least 7 milliliters of binding buffer. We have the best experience with 10 milliliters. The binding buffer is the most expensive component in this extraction protocol, besides the stupid tubes. But this is something that you have to kind of think through for yourself. What is the sample worth it? Can you play around with these things? And ideally, obviously, you would have a large pipette with which you can pipette 10 milliliters, but it actually works quite well with just pouring. But you have to be a little bit careful not to do too much, eh? And
that's the first part is
the first part. Oh, yeah. There we go. And that's why I have paper everywhere. And obviously also what we do, so these buffers, they can be kept for at least a month, I think, in the dark. But if you did use it, we'll label it as used, so that we know that it has been opened and has been, in case we trace any contamination here, that we never use the same aliquots that we have used to produce the contamination in the first place. Just like everything that is in the lab has to be completely DNA, well, as much DNA free as possible, right?
This cleaning is especially important because during library prep you're doing amplification, because you only have very low amounts of DNA in your sample, so you amplify your sample. So if any contamination would be introduced now, you would also amplify your contamination. So even though it's a very small amount of contamination you introduce here, you're going to amplify it and it will be overrepresented in your final library. So that's why it's important to try to prevent to get any contamination.
So you add your extraction buffer, you mix it by inverting, and move on to the next sample. And here you'll see, I don't know if you can see from here, but there's a bone precipitate here, well, like tooth in this case. And then obviously I'm not pipetting this out, I'm going. So what Tom is doing is preparing this tubes from the Roche kit.
Special tubes?
Yes, yeah, you can explain perhaps a little bit.
So the reason why we use this tube is because we use a lot of binding buffers. So in this case, we have the 10 milliliter plus the 1 milliliter extraction buffer, plus the 400 microliters of sodium acetate that are all in this tube now. So that's a volume of around like a little bit less than 12 milliliters. And usually, if anyone has used the spin columns before, like the Minolute or the quick spin columns, you can only add about like 1.5 to 2 milliliters in the columns. So the reason why we use these is because they can handle actually large volumes, so they go up to I think 20 milliliters even. So it's a bit unfortunate because they're very expensive and they're not so extremely special. Like yeah, they can handle more volume, but that's about it. That's the main difference. But that's the reason why we use these.
Okay. So what we're going to do now is we're going to add our mixture of extraction buffer and binding buffer into these columns, and we do it by pouring, right?
You save some pipette tips.
Yeah.
It does. Well, if you do have large pipettes, right, the ones that can handle 10 milliliters,
that would be ideal. Yes.
That's ideal. And what we need to do is to get all of this binding buffer through the silica membrane. So we centrifuge. And the centrifugation happens then. You have to have a centrifuge that can actually handle these, right, in your lab. So sometimes this is also a little bit tough to find in an ancient DNA lab or archive lab, but it would be good to have it. The fluid passes through quite well, but because they're tilted during the spinning process, you have to turn them around to make sure that all of the fluid has passed through. So what I'm going to do is I'm going to turn them by 180 degrees and spin them for a little bit more. Okay, so like I'm just turning every single tube by 180 degrees. And spin them for 2 more minutes. So what we do now is all the fluid has passed and it's collected down here. Obviously what you don't want to have is the fluid reconnecting with your spin column, right? That's not a good idea. But so what we'll do now, we will take out these receptacles here and put them into collection tubes. We don't need this anymore, but we need whatever is bound to the membrane. And this way.
And so you also have to be careful that the silica membrane is on the bottom of the tube, and this is actually what binds the DNA. So if you touch something by accident with it, you will bind all the DNA that it touches. So be careful if you take them out and put them in the tube that you don't by accident touch your own hand or anything. Then yeah.
It's an ingenious way of how to get rid of them. I'll show in a second. So all of the studies that, and it's always a good practice, keep your blanks throughout the entire process, sequence your blanks, because many people assess them and then they don't sequence them. But sequence your blanks, and good studies, they report the level, the number of fragments they recovered from the blanks, whatever there is, and usually it's orders of magnitude of difference between the actual extract and the blank. So there is a little bit of stuff that came out, and that would be a good reason to use a different collection. Now we put these things in, so now what we're going to do, we're going to wash them. And we're washing them with PE buffer. Whenever you get it commercially, don't forget to put in ethanol, right? Because it's an ethanol-based buffer, otherwise you're going to have a problem. And we wash them 2 times with 400 microliters each.
450.
450, sorry. And wash buffer usually drips out quite quickly. And also, you know, good practice in ancient DNA lab, try to touch everything as little as possible. So like not touch the rim of the tube with your pipette tip, not touch the rim of whatever else with your pipette tip. Don't breathe, don't live. And you take a new collection tube, right, and you put your things in there. I'm just putting them into a new collection tube and performing the dry spin, and I think it's going to be faster like that than if I move them back and forth.
No stress.
Right. And it's maximum speed for 1 minute, is that correct?
The dry spin, yes. Yeah.
And 1 minute. All right. Cool. We're almost there. So what we're going to do next, after the dry spin is done, we're actually taking siliconized tubes. And the idea behind siliconized tubes is the same as the idea behind tween 20. So siliconized tubes are, it's either siliconized or low bind. You can choose whichever you want. They both have the surface that prevents DNA from binding to the plastic. We are using siliconized tubes and tween in our elution buffer to make sure that the DNA stays in the solution and doesn't get absorbed by the plastic. So these are the tubes that you're going to be using for long-term storage. So this is why it's so important that they're siliconized. We also keep our bone powder in siliconized tubes for the same reason, that we don't lose any DNA to the plastic. So just putting like these are the collection tubes. I'm throwing them out, and I put the sample into the siliconized tube. I'll just plug the column into this. And again, you know, all of these steps now, they're quite critical, because the tubes in which your DNA is going to be, they are the ones with open lids. Right? So whatever happens to come on top of the lid is going to end up in your tube afterwards. So better not to do anything. Yeah, that's right. Okay. Yeah, so we have prepared the TET buffer, or TTE buffer, or whatever you call. So it's the tween buffer, tris buffer with tween 20. That's why the second T is there.
And then, so what we have been doing is we have been always doing eluting in 45 microliters, and then doing a second elution on the same column, which we call elution B. So elution A and elution B. So elution A obviously has a lot more of your DNA, and elution B still use a little bit. Sometimes if you want to do deep sequencing and you need a lot of material, you need actually your elution A and B as well. So there's also people that just elute 45 microliters and then another 45 microliters in the same tubes, so that you have 90 microliters in one tube, and this is a homogenized mixture of your elution A and B.
And what is the time of elution? I imagine the second time you elute, you leave it longer.
We didn't actually, no. So it might be maybe a better strategy.
No, I'm just asking.
No, so far we have been getting okay results, but we have seen that elution A contains a lot more DNA.
Logic, logic.
So that's right. Okay, the spinning goal.
1 minute at maximum.
At max. This is also. I'm not going to put them at maximum. I'm going to put them at 11,000, just because these lids, they need to remain open and they will go flying if you do it at maximum speed.
I can do that if you want.
So the extract is here at the bottom.